Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss DNA. We will have an overview of DNA that is relevant to healthcare professionals as well as healthcare students. Just as a reminder, it's very important for the healthcare student or professional to always look at the big picture. So although biochemistry tends to focus on very specific details, it's important for us healthcare professionals to look at the big picture so that we can actually apply this information to the well-being of our patient. So where is DNA located in the body? Remember, we have 200 different types of cells. So that's about 30 trillion cells. And we mentioned in an earlier video about 80% of these cells are red blood cells. So that's about 24 trillion simply red blood cells. So DNA is located in the nucleus. You have your 46 chromosomes, your 23 pairs. And then there's some DNA, of course, in the mitochondria. So almost all the 200 different cell types in our body has DNA, except this very important cells, your red blood cells. So the 24 trillion red blood cells, your matured red blood cells that are floating around in your bloodstream, they do not have DNA. Okay, by having DNA or organelles, these red blood cells will become very heavy, and that would be very difficult for our heart to pump all these red blood cells across our circulation. So how do we form DNA? The basic chemical reaction is a reaction between nucleotides. The 3' hydroxyl group present in one nucleotide will attack the phosphorus nearest the sugar of another nucleotide, and that would cause the two sugars to join together by a phosphodiester bond. And now we will get two nucleotides joined together, or we can call it a dinucleotide. And then we have again the free 3' hydroxyl group of a nucleotide in the dinucleotide, and this hydroxyl group can of course attack another nucleotide to form trinucleotides, and that trinucleotide will again have a 3' free hydroxyl group which can attack another nucleotide phosphorus and form another longer nucleic acid, tetranucleotide, pentanucleotide, and so on. And that's how we get long nucleotide polymers that we call nucleic acids. And the two major nucleic acids are DNA, which we'll talk about today, and RNA, which we'll talk about in a future video. It's also important to always remember that once we start talking about longer biomolecules, we need to identify the direction. So the sugar that has a free 3' OH, that's called the 3' terminal. Then the other end of this molecule will be the 5' terminal. Okay, it's all very important to remember that sequences, because these nucleic acids basically carry information, and this information is in the form of nucleobases. So the information has to be written in a certain form, and we have to follow the rule that this information is always written from the 5' end to the 3' end. 5' end. So you can see the 5' end is an A. So we have our A. And then the 3' terminal of this dinucleotide is a T. We have our T. And so the sequence of this is AT. So sometimes we write 5' AT, 3'. Most of the times, if you do not see the numbers, it's understood this is your 5' and your T would be your 3'. So when we actually make DNA, and that happens, for example, when the cell divides or when the DNA has to be repaired, it's damaged and it has to be repaired. So these are two examples of when we actually make DNA. We use a number of different enzymes. The most important is called DNA polymerase. This enzyme basically uses one of the two strands of DNA. Remember, DNA has two strands. So it uses one of the strands of the DNA called a template strand. And then it uses this rule, this pairing GCAT, and the template strand is copied into the new strand, T into A into T, G into C, A is copied into T, and so on to form the new DNA strand. So that's how we make our DNA. Let's look at the basic anatomy of DNA. So basically you can see we have a sugar phosphate backbone. So we have the sugar, we have the phosphate, again the sugar, phosphate, and so on. Same thing on the other side. The other strand, sugar, phosphate, and so on. And then attached to the sugar are the nucleobases. These inside the DNA is in the interior of the DNA is the nucleobases. So the sugar phosphates on the outside, as shown here, while the nucleobases are on the inside. So you can see the nucleobases on the inside, the nucleobases, while the sugar phosphate is on the outside of the DNA. And you can see that when you see it from here very clearly. The other important features about DNA that we should keep in mind is the fact that the DNA is double-stranded. We have two strands. We have one strand here and we have the other strand there. And these two strands are complementary to one another, which means if you see a G on one strand, then the opposite strand will have a C. If you see a A on one strand, the opposite strand should have a T. So it always follows these ATGC rules. These two strands of DNA are anti-parallel, which means one strand runs in one direction, 5' to 3'. The other strand runs in the opposite direction, 5' to 3'. The most important part of this DNA is information, and this information is stored as nucleobases. So we talked about this in previous videos. DNA is a very long molecule, the 46 chromosomes. The DNA in the 46 chromosomes very, very long, and so all of these chromosomes have to be stored in a very small space that we call the nucleus. So there is a lot of folding of the DNA double strand. So DNA is wound around basic proteins. These are proteins that are positively charged. So they are wound around basic proteins called histones to form a basic structure of a chromosome called a nucleosome. So nucleosome is basically DNA wound around a set of histones. These nucleosomes then further fold, become more compact to form fibers, which themselves fold to become filaments, and these further fold to form the chromosomes. So there's a lot of folding of your DNA to pack into the chromosomes, and then all these 46 chromosomes we need to pack in our small space that we call the nucleus. So a lot of folding of DNA strands into chromosomes. What are the forces that stabilize DNA shapes? So one very important force is hydrophobic. Remember, everything is surrounded by water except for the membranes. All parts of the cells are surrounded by water. And so is DNA in the nucleus. The water forces things that are not water soluble, the non-polar part of the DNA, which is your nucleobases, which are aromatic and non-polar. So it forces the nucleobases to be inside the folded molecule. So that's called the hydrophobic force. The second force is called electrostatic. So here we have our phosphates. Phosphates are negatively charged. They interact with something that is positively charged, either magnesium ions or histones. Histones have a lot of positive charges. These are what we call basic proteins. Basic proteins are not that common in our body. We'll talk more about this under proteins, but some basic proteins such as histones play a very important role in our body. Most of proteins are acidic proteins, but we'll talk about this under proteins. So this interaction between the phosphates of DNA and the cations such as magnesium or basic proteins such as histones, we call this electrostatic. And that helps stabilize the DNA shape. Another important force that stabilizes the shape is what we call stacking interaction. These nucleobases are aromatic, so they are polycyclic aromatic compounds, and they are unsaturated, so they have pi bonds. And so the pi electrons on one base can interact with the pi electrons on another base. And this interaction is called stacking interaction. Stacking interaction plays a very important role in stabilizing the DNA shape. The fourth force that stabilizes DNA shape are the hydrogen bonds, and this happens between the nucleobases on one strand and the nucleobases on the other strand. So you can see hydrogen bonds form between nucleobases on opposite strands. The GC interaction involves 3 hydrogen bonds, while the AT interactions involve 2 hydrogen bonds. So clearly those parts of DNA that have a lot of G and C are stronger because there are 3 hydrogen bonds per GC compared to 2 per AT. So because of these four forces, hydrophobic, electrostatic, stacking, and hydrogen bonding, DNA has a very stable shape. And these forces, we'll also talk about these under proteins, except for stacking interactions, which we rarely see in proteins. The other three, hydrophobic, electrostatic, and hydrogen bonds, are also very important in stabilizing the shapes of proteins as well as many other biomolecules we find in our body. So, one very important question, how DNA structure helps perform its functions. So in the previous videos on lipids and carbohydrates, we talked about how is the chemistry of that chemical or the biomolecule, how is that related to its biology? Okay, which means its function, so what it does in the cell. That's a very important question. We sometimes call that the structure for the chemistry and activity for the biological function. We call this a structure-activity relationship. So what is the structure, SAR? What's a structure-activity relationship of DNA? What's so special in the DNA structure that allows it to do all the functions necessary for the cell? You can see from this diagram how important the information in DNA is. This is your information in DNA, the sequence of bases, nucleobases in the DNA. So one of these strands that we call the template strand, template strand, this information in your DNA template strand is copied or transcribed by a process that we call transcription into various forms of RNA. This form called messenger RNA actually carries the information for the primary structure of the protein, while the other forms like tRNA and rRNA help in protein synthesis, while other RNAs help regulate gene expression. So once these different RNAs are made, they undergo processing, and we'll talk about this later and in a future video. And once the RNAs are processed, they are transported out of the nucleus into the cytoplasm. So all proteins are made in the cytoplasm. Now here the nucleotide language of the mRNA. So this is your mRNA. This is a nucleotide language, right? DNA is nucleotide language. RNA is nucleotide language because they're made of nucleotides. So this nucleotide language has to be converted into the primary structure of the protein, which is amino acid language. Proteins are made up of amino acids, so the language of proteins is different. So protein synthesis is therefore called translation. We are translating nucleotide language into protein language. And this follows the rule of the genetic code, which we'll talk about again in a future video. Once the proteins are made, they undergo protein folding, there might be processing, and then they are transported to the right location in the cell, whether they're on the cell membrane or in the cytoplasm or in the nucleus and so on. And these proteins form what we call the protein profile of the cell. So all your 200 different types of cells have different protein profiles, and that's why they have different cellular activities. That's why they are specialized. And all of these specialized 200 different types of specialized cells will then carry out their functions necessary for our survival, our anatomy, and physiology. So you can clearly see how important DNA information is that indirectly affects our anatomy and physiology. Very important relationship, a big picture to understand for healthcare professionals. So clearly, if something goes wrong with this information, it might end up affecting one or more proteins in the cell, and that cell, a certain specific cell, or maybe more than one specific cell that shares the proteins, they might be affected, and that might lead to changes in our physiology and our anatomy. And if they are significant, then that might lead to pathology, to a disease. Okay, so very important. You can see the relationship between this DNA information, proteins, and our anatomy and physiology in health as well as in disease. So this information is critical. Therefore, it must be a very stable form of information, and it must be protected, because in our cells there's all kinds of chemicals and especially free radicals. Free radicals are very, very reactive species, chemical species that can form from radiation and many other sources. So, and these free radicals can attack anything, and if the DNA is nearby, they can attack the DNA and change this information, and that can lead to all kinds of pathological problems. So very important that we need to have a stable and well-protected information. So what are the features in DNA that makes this information stable and is well protected? Well, one is the nucleobases, which is the information, is hydrophobic and it's buried away from water. So it's inside. It's in the inside of the DNA. The sugar phosphate is on the outside. And the sugar phosphate actually helps protect the nucleobases from water-based free radicals. Okay, a lot of very dangerous free radicals, such as the hydroxyl radical, is water-soluble. So this sugar phosphate backbone is basically helping protect the nucleobase information inside. The nucleobase information, nucleobases, as we mentioned, are aromatic. Aromatic chemicals are very stable. So their nature, aromatic compounds of the nucleobase, the nature of nucleobases being aromatic, increases the stability. Another very important feature of DNA is, remember, DNA is deoxyribose. The D stands for deoxyribose. Deoxyribose. So you can see there is, on the sugar, you can see there is no OH here. What if there was an OH? If there was an OH here, this OH could attack this very near phosphate on the backbone and break this phosphate. Okay, so the OH, if present, can actually break this phosphodiester linkage. So by not having the 2' OH in the deoxyribose, it makes the DNA backbone more stable, increases the stability. The opposite happens in RNA. RNA is a chemical we don't need to be very stable. So you actually see there's a possibility in RNA that the 2' OH can break the RNA backbone. So DNA is to be stable, we don't have 2' OH. Deoxyribose. Another very important feature of DNA now, DNA has to store a lot of information. Okay, a lot of information, and all this information has been stored in a tiny space that we call the nucleus. Now, the nucleus is about 2 micrometers in dimension. While the 46 chromosomes, if you extend all the DNA out of every chromosome and join them together, they are about 2 meters long. So there's a lot of DNA to be packed into a very, very tiny space. So what is the structural features of DNA that allows it to be packed within a tiny amount of space that we call the nucleus? One is the nucleobases themselves. The information is planar. These nucleobases are planar. They are flat molecules. So they look like the pages in a book, two-dimensional. And therefore you can stack the pages just like a book. The other important feature of DNA is that it's negatively charged, and that negative charge allows the DNA, so if this is your histone, your DNA can actually wind around the histone and allow tight packing. By winding around histone, we can pack more DNA. And then the histones and the DNA, they form what we call nucleosomes. The nucleosomes, they themselves undergo further folding to form fibers, and then filaments undergo further folding to form these chromosomes, which are very, very tightly packed DNA. Another important function of DNA, it carries information, but this information must be available only to specific cells, and that too at appropriate times. So remember, we have 200 different types of cells, so except for these red blood cells that don't have DNA, the rest of the cells have exactly the same 46 chromosomes. Okay, so all your 199 remaining cell types have exactly the same 46 chromosomes. So basically they carry the same information. Okay, they carry the same information. However, the information available to one cell in some cases should not be available to another cell. Okay, so some information can be shared by all cells. Okay, that's fine, but some information should be only available to certain cells. So for example, the beta cell, pancreatic cell in your pancreas, the information for the insulin gene, this insulin gene information should only be available to the beta cell. That's the only cell that makes insulin. It should not be available, should not be accessible to the neuron or to the muscle cell or some other cell type. Okay, so it's very important that something in the DNA must allow this information, certain information, to be accessible, while other information should not be allowed in the for a certain cell type, and this information should be available under appropriate times. So how is this possible? How is the same 46 chromosomes in various cell types, the same information, why is it that some information is available only to certain cell types? Let's see how this happens. So remember, the DNA is wound around histones. So this means that a lot of information, let's say there is a gene information here, this information can only be available if we remove the histone from that DNA. And this access to certain genes is only allowed in certain cell types. So if this was the insulin gene, this access to the insulin gene by removing this histone here is only happens in the beta cell of the pancreas. The other cell types do not have the protein machinery or other biomolecules necessary to do this, to get access to this insulin gene here. Only the beta cell of the pancreas has access has the necessary machinery to get this information. So by restricting information in different cell types, what the body does, it allows specialization or differentiation of all the 200 different cell types. Okay, that's why we have differentiated cells. That's why we have the 200 specialized cells, because they all have access only to certain information in their DNA. So how does let's say the beta cell wants to get access to its insulin gene? And how does that happen? Well, this is called chromatin remodeling. To get information, the cell needs the necessary enzymes and other proteins to get access to this gene information. So one example of chromatin remodeling is that histones are positively charged. They're very rich in positive charged amino acids such as lysine and arginine, and we'll talk about this proteins later in a later video under proteins. And because of this positively charged amino acids, there is this amine groups that can be protonated and therefore positively charged. Phosphate is negatively charged. So the histones and phosphate can form very tight complexes. So to get access to this insulin gene, basically there are proteins such as this enzyme called histone acetyltransferase that can take an acetyl group and add it to the amine. Now that amine becomes an amide, and amides do not have positive charges. So this is a acetyl group. This is an acetyl group. So by acetylating the amine, the positive charge is lost, and now the DNA is no more bound to the histones through electrostatic. The positive charge on the histones are gone, and the gene is available. The gene is accessible. That gene is then transcribed into mRNA, let's say the insulin gene, and then that insulin gene is then translated into insulin. So this accessibility is only, let's say, allowed in the beta cell of the pancreas, and that's one of the specialized functions of the beta cell. It's the only cell type out of 200 cell types that produces insulin. So in a similar fashion, the albumin gene is only accessible by your liver cells. Like it's only accessible in the liver cells. Hepatocytes. Liver cells are the only cells, the hepatocytes are the only cells that can make albumin. Okay, so many genes are accessible only to certain selected cell types, while some genes such as your, let's say, glycolysis enzymes, we'll talk about this later in the metabolism, all cells can do glycolysis. So the gene for your glycolysis enzyme should be accessible by all cell types. Okay, so that's how the 200 different cell types in your body have selected protein profiles. They all have very different protein profiles, and that protein profiles allow them to carry out their specific functions for the human body. Another very important thing that DNA should do is that all the information in the DNA, the 46 chromosomes, must be copied exactly during cell division. Okay, so when a cell divides, all this information must be copied so that the new cells also have the same DNA information. So how this is done, what's so special in the chemistry of DNA that allows copying this information? One is that there are two strands, they are complementary, the information is complementary. And the second important feature is that these strands are held together by weak forces. So these two strands can be separated. They can be separated before the cells divide. And then this strand can be copied and this strand can be copied, as shown here on the right hand side. So here's your original DNA. The two strands are separated. This strand is separated from this strand. And then the information is copied. C to G, A to T, T to A, on the new strand is made. And similarly from this strand, another new strand is made. So this original strand, the new strand goes to one cell, while the new and the original strand goes to the second cell. So the new cells also get the same chromosome information as was contained in the parent strand, parent cell. Another good thing about having two strands is what if radiation damage some of the DNA? Remember, DNA is such critical information. So what if we lost this information here? Well, there shouldn't be a problem because the other strand has the complementary information. So we can always put back the T here. The cell knows that this lost information here would have been an A because it's next to a T. Okay, so we have a lot of very efficient DNA repair systems, which we shall talk about in a future video. So damaged information must be recovered, and having those two strands help us do that. Okay, now that we know what our DNA has to do and how it does it, now let's look at what is the information in our DNA. Okay, a lot of this work is still been going on, and there's a lot of this information came out more recently. So if you're interested in knowing about all the information in the 46 chromosomes, there are two databases. One is called RefSeq. It's maintained by NCBI at NIH. And the other one is maintained by a European Molecular Biology Lab, and that's one of them is called Ensemble, and the other one is called GenCode. So what is this information in our DNA? Again, the big picture is very important. There's a lot of definitions in this, a lot of terms that are used for this information. It can be very, very confusing. But again, for healthcare professionals, it's very important to look at the big picture. So when you look at the big picture, let's focus here. 2% of all your DNA is what we call coding DNA. This is the DNA that contains information to make the primary structure of the protein. Okay, so the protein coding genes, this information is eventually made into proteins, and then that forms the protein profiles of the 200 cell types, which give us the anatomy and our physiology. The remaining 98% is used to regulate the protein profiles of cells. So the remaining information makes all kinds of RNAs, and these RNAs can regulate at a level of transcription or RNA processing or translation or all different steps of protein processing and so on. Okay, so basically this is what our important information is. The rest of the information is used to regulate our protein profiles. And some of the other information is also used to make certain shapes in our DNA, such as centromeres, which are very important during cell division, and telomeres, which are the ends of chromosomes, which we'll talk about in a few seconds. So some important definitions for healthcare professionals. We talked about protein coding genes, and we'll look at introns in a few seconds. The rest of the information on your DNA includes things like promoters, enhancers, silencers, And then we have genes called long intervening non-coding RNA genes, or LNC RNA. Then we also have small non-coding RNA genes. And then we have what we call pseudogenes. These are non-functional genes that resemble protein coding genes. And then there is telomeric DNA, which is repetitive DNA. And then centromeric DNA, which is also a repetitive DNA, also called satellite DNA. So centromeres DNA gives structures to the centromeres, part of the chromosomes, which are very important during cell division. This is where the spindles attach to transport the chromosomes to the two different parts of the dividing cell. And then we have the telomeric DNA, which forms the telomeres, and this is very important. We'll look at this later under cell division, under DNA replication. This is to prevent the ends of the chromosome from becoming too short and then losing some of the essential genes. So the genome is our 23 chromosomes plus the X or the Y chromosome. That's all in the nucleus, and this is what your nuclear genome is. And then we also have mitochondrial DNA. So in your mitochondria, you have a circular DNA, which about 17,000 base pairs, and there are about 37 genes found in this circular DNA. 13 of these genes code for proteins, and the other genes code for RNA. The rest of the proteins needed for the mitochondria will come from the genes from the nucleus. Okay, since proteins carry out most of the activities of our 200 different cell types that lead to our anatomy and physiology, let's have a quick overview of how is the flow of information. How is the information in your DNA used to make the primary structure of a protein? So here, for example, is the template strands. Remember, only one of the two strands is copied. The template strand is copied. The non-template strand is not copied. So here's the template strand. Here's the information, TAC. T becomes A in your mRNA. A becomes U. C becomes G, and again so on and so on. A becomes U and so on. Okay, so this whole thing is copied, and then we have the intron. So out of your 20,000 genes, about 600 genes do not have introns, but the remaining 19,000 genes do have introns. So the intron is also copied in your mRNA during transcription. And this is done by the enzyme RNA polymerase. It copies your DNA, so your exons and your introns are all copied into the mRNA by RNA polymerase. The next step is RNA processing. Many things happen in RNA processing. We'll talk about this later. But one of the things that happens is the intron is removed. The intron is removed, and then the exons, this exon is connected to this exon, right between the C here and the G here. So you can see the two exons connected together. And then that RNA is further modified. For example, a cap may be added, a poly A tail is added, and then it's transported out into the cytoplasm. And once in the cytoplasm, it then uses the rules of the genetic code. The genetic code, which says AUG in the mRNA, will be translated into methionine. The amino acid methionine. UUU will be translated to phenylalanine. CCC into proline, GUU into valine, AAA into lysine. And then once it hits UAA, which is what we call a stop codon, protein synthesis stops. Okay, and that's how we get our proteins. So the information in DNA, this information in your protein coding gene will be used to make the primary structure of this protein, which will then fall into a certain shape, secondary structures, tertiary structures, which we'll talk about under proteins. And then this protein will carry out its function for the cell. So this is how the information flows from your DNA to the protein. When you look at the cell, the nucleus is the place where a lot of RRR, ribosomal RNA, is made. And then you can see clear areas in the cell. We call that euchromatin. And this is the DNA that's actually being transcribed, that is actually being made into some sort of RNA. Okay, while the darker areas in the nucleus, very close to the nuclear envelope, this is called heterochromatin. This is a DNA that's not being transcribed. So this is a DNA that has a lot of histones attached. The histones are strongly attached, and so this area is very compact. It's what we call heterochromatin. While this area, a lot of histones have been removed and is transcribed into RNA. So before we end this video, a few words about repetitive DNA. These are DNA sequences that are repeated, and they could be in two types, tandem repetitive or interspersed repetitive. So in tandem, the repeated sequences are next to one another. The repeated sequences are next to one another. While in interspersed, the repeated sequences are not next to one another, not adjacent to one another. They are interspersed. They can be anywhere along the chromosome. A few words about tandem repetitive DNA. They include satellite DNA, which is important for assembly of centromeres, mini satellites, which are smaller sequences, for example, found in the telomeres, and they're important for DNA fingerprinting. And then there are even smaller repeat sequences called microsatellites. And a lot of diseases are associated with microsatellites. For example, Huntington disease, the gene HTT has a CAG microsatellite that is repeated more than 30 times. Usually for normal, in undiseased patients, this number is less than 30. But once these repeats become more than 30, it's associated with the disease. Similarly, in certain forms of muscular dystrophy, the androgen receptor gene, or AR, has a CAG microsatellite that is repeated 38 times, and normal is 36. In myotonic dystrophy, the gene DMPK, it's a protein kinase gene, which we'll talk about later, has a CTG microsatellite that's repeated more than 50 times, while the normal is less than 34. In Fragile X syndrome that affects the brain, the CGG triplet in the FMR1 gene is repeated many times. Okay, so microsatellites are used, besides the association with diseases, they're also used for paternity testing and forensics. A lot of your ribosomal RNA genes are also tandem repetitive sequences. And then finally, a few words about the interspersed repetitive sequences. These are kind of interesting because these, what we call as transposable elements or transposons, these are jumping genes. They actually can jump from one part of the DNA to another part of the DNA to another part of the DNA. So they actually move around, which is kind of fascinating. They move around in your DNA, jumping genes. And they are believed to increase the genetic diversity, they affect chromatin structure, and they also regulate transcription factor binding. So a lot of these this information is quite new. So I'm sure a lot of this information will find a lot of fascinating things about this repetitive DNA and their functions. So there are two types of transposons. There's a class 1 and a class 2. So in class 1, basically the transposon makes its RNA. This RNA is then reverse transcribed by an enzyme called reverse transcriptase. And then this sequence is embedded back, reintegrated back into the DNA by another enzyme encoded by the transposon called endonuclease. So in this fashion, by copying its DNA, its sequence, and then inserting it somewhere else, this gene has jumped to another part of DNA. And examples of these class 1, what we call retrotransposons, include long interspersed nuclear elements or lines, as well as short interspersed nuclear elements or signs. And an important sign that is medically important is called the ALU repeat, ALU, and that is connected or has association with a variety of diseases, including breast and ovarian cancer, hemophilia, and neurofibromatosis. All of these have been associated with ALU. The second type of transposon, DNA transposon, is called class 2. So in this case, this transposon encodes the mRNA, transcribes the mRNA, and this mRNA then makes a protein, a multifunctional protein called transposase. And what this protein does, it recognizes sequences on either side of this transposon and it cuts it. Okay, so it actually removes this transposon, and then the enzyme now attaches it, pastes it on another part of DNA. So basically, the same enzyme transposase will cut the transposon out and then reattach it somewhere else. We call this the cut and paste mechanism. Cut and paste mechanism. And in this way, this transposon is jumping to another part of the DNA. Okay, so tandem repeats and interspersed repeats. Okay, so I think that's the some of the important definitions for this video. Let's stop here. So this is an overview of DNA that is relevant to the healthcare professional as well as the healthcare student. We will see you in the next video. Have a good day.